We wait a few more seconds and people are coming in. And then we are live in YouTube as well. Perfect. So here we go. So hello, everyone. It's my pleasure again to introduce the speaker of today, so Peter Schaffer. So as usual, I go through a very brief introduction and going through the CV a bit. So Peter did his PhD at MBL Heidelberg. With a PhD thesis, actually, that makes a lot of sense also for what he has done afterwards, I think, a bit on the mechanism of apical membrane trafficking polarized epithelial cells. And after that, he moved for a postdoc at the University of California and San Francisco and University of California, Berkeley, with as a supervisor, Marc Tessier-Lavigne. So after that, after this postdoc, he moved to Columbia University in 2001, where he accepted and got a position as assistant professor. And then in 2008, or since 2008, is professor at the Biocentrum at the University of Basel in Switzerland. So Peter is very much interested in the studying the formation of neuronal network. And in particular, I would say that in the last years, he has been a pioneer in understanding and studying how neuronal adhesion molecules contribute exactly to the establishment of those neuronal networks. And he has been doing this as a very basic research, so in let's say a control condition, normal conditions, but also how those adhesion molecules contribute, or mutation in those adhesion molecules contribute to disorders such as autism. And some of those molecules are the neuroligin and neurexins. And so, as you probably, most of you know, those are heavily involved in those autism spectrum disorders. So I think today Peter is going to tell us something that he has started already some years ago about alternative splicing and how this contributes to the specification of those neuronal networks. And so, Peter, we are very much, certainly I am very much, I'm sure everyone else looking forward to your talk. So I think you can share your screen now and start your presentation. Thank you again for being here.
Yeah, thanks, Gaia. It's really a great pleasure to give this seminar today. And I'll share with you essentially two stories in the lab, which are sort of the two pillars in the lab. One is on RNA splicing, and the other is actually transferring some of the knowledge that we gain in the lab on normal nervous system development to understanding pathophysiology of autism. And just as a means of introduction, there are many different ways of looking at circuits, and I think these days circuit neuroscience is really very en vogue, and what most people really study is to see, either to map out what the connectivity matrix is, and then how the activity of different cells drives certain behaviors, how they are modified during learning processes, and so on. What we are largely focusing on is in a way much more cell biological, and it is slightly different. And that is to understand how you actually build circuits of the desired properties. And what we focus on is largely the synapses, because if you look at synapses across different classes of neurons, they're actually hugely different from each other. They're not just nodes where cells connect to each other, but they're really vastly different with respect to morphology, their strength, and also their functional properties. And I've just taken here this cartoon from the hippocampal wiring diagram, and one EM picture section across the Schaffer collateral synapses. And if you just compare that to one of these mossy fiber terminals here, you see that they're really vastly different. One is tiny, the other one is huge. One is hundreds of vesicles, the other tens of thousands. The numbers of release sites are completely different from each other, and also the forms of plasticity that they undergo. And what we know is that if we look across different organisms and we look across different individuals from one species, is that this really happens highly reproducibly. So there is something like a blueprint that instructs forming synapses like that of those defined properties. And we also know that this is really fundamentally important for what information and how that circuit computes information, and also then how that circuit can actually drive behavior. And we know that in part from studies on the synapse itself, and then also from studies on disease states, because a lot of these molecules that are thought to instruct the specification of the synapses, when they mutated, result in severe disease states. And so as a sort of a guiding model that we're pursuing is that we think of this to a large extent as being driven by something like a molecular code, meaning that particular classes of cells would display at their synaptic surface certain molecules, adhesion molecules, but also other signaling molecules, and that these, by finding appropriate partners on the transsynaptic side and the other side, would form modules across the synapse, and that then the combination of these different modules leads to some interpretation that are the particular synaptic properties that are desired. And by arraying molecules and their interactors over different classes of cells, you would then essentially code for these basic functional properties of the synapses across the surface of this recipient parameter cell, for example. And what I want to do today is share with you some work on alternative splicing that we've done, where we've really pursued the idea that a big part of this molecular code could be driven by RNA splicing and its regulation in different cell populations. And this is work done by two PhD students in the lab, Lisa Traunmüller and Elisabetta Folanis, in collaboration with the postdoc Andrea Gomez. And then in the second part of the talk, I'll turn sort of from the normal development to the disease relevance, where we've then essentially used one of these synaptic cues that are instructing certain functional properties on synapses and cells, and placing them in a circuit context to ask how mutations in this gene could possibly be responsible for certain aspects of autism-related behavioral alterations. And that second part is work that was really largely driven by a single postdoc in the lab, Hannah Hörnbach. So why study splicing? Why would we do this? And it's maybe rather trivially motivated here, but if you look evolutionarily across organisms and tissues of different complexity, of different phenotypic complexity, it's become clear immediately when genomes were being sequenced that this is very unlikely to be driven primarily by the number of protein coding genes, but has been immediately hypothesized to be somehow dependent on transregulatory elements such as enhancers, transcriptional regulators, and also post-transcriptional regulators, regulation of splicing. And that had been postulated to possibly be relevant for making the brain of a mouse so much more sophisticated and complex than these 302 neurons that are in a very effective way wire up and encode behaviors in C. elegans. And then further sort of genome-wide studies have added support to this idea. And this here is taken from a paper from Ben Blanco's lab, where they looked across alternative splicing in different organisms. So you see a human, macaques, mouse, all the way down to the frog, and looked at the relative abundance of alternative splicing events. And they also looked at that in different tissues. And really the two things that you can see from that are really striking is that one, if you go from human, if you go out an evolutionary distance, then the extent to which alternative splicing is used is strongly decreasing. And the second thing is that if you look across these tissues, then it becomes clear that it's really the nervous system that makes the most use of this alternative splicing, and thereby putting forward the idea that possibly this could have something to do with the complexity of the tissue and being able to create this phenotypic diversity that you need when you build a complex nervous system. So this was really from an evolutionary perspective, the motivation for us to be interested in this, and to ask whether alternative splicing could actually be an important contributor to synapse specification. And the second motivator to do this had really come from a class of molecules, adhesion molecules that we'd been studying for many years, and these were the Nexns. And they are largely presynaptic molecules sitting here and building complexes with various binding partners in the postsynaptic side. And already when they were discovered by Tom Sudop's lab, it had been recognized that they're highly diversified by splicing. And this is illustrated here. So there are multiple genes for these Nexns. There are multiple alternatively spliced segments across them. And by permuting the different exons that can be incorporated here, so by the splicing process, either being skipped or being incorporated into the transcript, you end up with thousands of different theoretical variants of these. So meaning you now have a molecule that's sitting at the synapse and that has the potential to really make it more than 1,000 different tags. We've essentially gone in and tested this experimentally and seen that indeed by long-read sequencing with PacBio methods, there are more than 1,000 variants detected in the mouse brain. And we've also seen that these variants are actually, certain variants are restricted to a certain neuronal cell type. So this really fits this idea that cell type specific splicing in some form could contribute to shaping synapses. And so what we then wanted to ask really is to test this. And now it's of course tedious if you go in and you want to knock out each single one of these 1,300 variants. And so we took a little detour to address this, to ask what's the functional relevance. And that is that we thought, well, if you produce specific isoforms in particular cell types, then there should be machinery that actually drives their production. So that a certain class of neuron, like this one here, would have one set of splicing factors, and this class of neuron here, for example, would have a different set, and so on, and that would shape the molecular makeup of these cells, and thereby possibly contribute to these transsynaptic codes. And so to test this, we essentially went out and looked for RNA-binding proteins that would be expressed not just in neurons or in neuron-specific way, but in certain subclasses of neurons. And one factor that I want to focus on in this part of the talk today is a protein called SLM2 here. It's one of a small class of proteins, and this is an RNA-binding protein that essentially recognizes sequence elements in an intron and can thereby steer whether in the splicing event you either incorporate or you're skipping a certain alternative exon. Now, there are actually hundreds of splicing factors. So why would we look at this one in particular? And the reason is really that we found that it's rather selectively expressed in certain cell populations. And you see this here with a view of the hippocampus, and the green labeling here is the labeling for this SLM2 protein in the nuclei of these cells. And what we find is that it's essentially only expressed in the pyramidal cells. It's completely absent from the dentate granule cells, and then it's also in a subclass of somatostatin interneurons. So it sort of matches this idea of being a factor that could create a certain cell type specific splicing program. So to ask whether to test this hypothesis, whether disrupting such a cell type specific splicing factor would produce synaptic phenotypes, we made knockouts in SLM2. And what I'm showing you here is data from a global knockout. So it's a global ablation of the protein, but keep in mind it's very selectively expressed in the first place. And I should say we looked at a lot of parameters. Many of them I will just not show you here, and they were completely unchanged. So synapses formed in normal numbers, the dendritic spines had normal morphology, presynaptic properties were unaltered. However, we found one thing that was really significantly changed, and that's the abundance of AMPA receptors at the synapses of these mutant cells. So this is looking at the Schaffer collateral synapses in the hippocampus, and when you record AMPA to NMDA receptor ratios, you find that these are elevated in the SLM2 knockout. This is evoked transmission that is elevated. This is looking biochemically, where you see GluA1 levels are elevated, whereas GluN1, so NMDA receptor subunit levels, are completely unchanged. Now this was really quite surprising because it was such a selective phenotype. And keep in mind, we're not mutating a synaptic protein, we're actually mutating a nuclear RNA-binding protein that regulates splicing. And so to now understand how the loss of this RNA-binding protein would result in changes in synaptic function, we then mapped the splicing program. And there's one more piece of data that I actually just forgot to tell you about. We also looked at long-term potentiation at the Schaffer collaterals, and you see that with the theta burst stimulation, you induce nice plasticity in the wild type, and there's a complete loss of LTP in the S1 knockout. So now the next step was to map the splicing program. And so we basically sequenced the very deep RNA-seq assessment of transcript isoforms in the S1 knockout. And what this shows here is a volcano plot of individual exons in the fold change in splicing index. So this would be what goes to the right, shows an increase in splicing index, so an increase in incorporation. What goes to the left would show a decrease. This is the p-value, and so you can see there is S1 itself altered, and that was really a big surprise. We really only found a handful of exons that were deregulated in these global S1 knockouts. And this was a surprise because there had been plenty of studies previously mapping splicing programs for RNA-binding proteins like the RB-FOXes or the PTBs and NOVAs, and they typically regulate hundreds of different exons. And here it seems this only regulates really a handful. And what makes this handful even more interesting is that 3 of them are our favorite molecules, the Nexns. So this is this exon encodes a 30 amino acid insertion in the Nexn molecule, and then to syntaxin binding protein 5, and that is a component of the free synaptic release machinery as well. So as if this RNA binding protein is really tailored to controlling synaptic properties. Now you could worry that we only find so few things because there's too much noise in our analysis, or we're missing things, and so we really wanted to test whether the regulation of these synaptic proteins is critical for the phenotypes that we observed. And so we decided to do a genetic rescue by restoring splicing pattern of one of the targets of S1. And so we focused on the Nexn1 gene because that's highly expressed in the CA3 neurons, the presynaptic partner in our physiological experiment. And what we had seen was that in the wild type, the dominant form of Nexn that's expressed is a form lacking this exon 21. And in the knockout, the cells produce an isoform that contain this additional exon. And so we reasoned that if this is really relevant for the phenotype, we should just knock out this exon, cross this mutant mouse with the S1 knockout, and then we should rescue phenotypes. And really quite remarkably, I think this worked a lot better than we expected even. We could rescue essentially, to a large extent, the phenotypes that we had described. So here, this would be basically the restoration in the cartoon form of this AS4 minus or exon 21 lacking forms in the double mutant. When we look biochemically at the AMPA receptor levels, this would be the wild type. This is the elevation in the S1 knockout. This AMPA receptor elevation goes back down to wild type levels. And we are also rescuing, at least partially, the theta burst induced plasticity of these synapses. And this really strongly argues that the splicing program regulated by this factor is not very, very broad, consisting of hundreds of transcripts that are somehow synergizing together, but that it's very, very targeted and controlling very specific molecules that then instruct the function of these synapses. Now, here, this is all hippocampus-centric. We actually also did an experiment that sort of should assess sort of the whole body relevance of this observation, and this is looking at behavior in the animals. As a disclaimer up front, clearly these are not behaviors that necessarily depend on the Schaffer collaterals, but what these experiments tell us is that the Nexn splicing is really critical, even if you use a whole animal readout. So this up here is an object recognition test that many of you are probably familiar with. So you give mice two unfamiliar objects to explore, then you replace one of these objects, and after 1 hour wait time, the mouse goes back into the arena. And wild-type mice are always strongly attracted to the novel object. And you see this here by this discrimination ratio. The SLM2 knockouts actually have a very interesting phenotype. They know what's new, but they make a different choice. They actually avoid novelty, and they're strongly biased to the familiar object. And if you now look at these double mutant mice where we restore this single exon, you're getting back the wild-type behavior. Same for hyperactivity phenotype. So knockouts are hyperactive, and the double mutants have normal levels of activity, and knockouts also have a marble burring deficit, whatever that means, and double mutants are rescuing that. And so what this really meant, or it made us think quite differently about how splicing could be instructing neuronal function, that there can be at least one, SLM2, very dedicated splicing regulators that very selectively regulate splicing, and in this case that this was really targeted to control synaptic properties and thereby the instructed function. Now this is something that we sort of stumbled into, and we're really surprised by the selectivity of this. And so next we really wanted to ask, well, was this just a lucky exception, or is this possibly a broader principle? So how similar and different are, if you now go down to specific neuronal classes, how similar are they in their splicing pattern? And does that carry information for how they form synapses and what the function of these synapses are? And you know that over the past decade or so, there's really been an explosion in the definition and wonderful convergence of information on how we can define cell types by lining up the morphology, electrophysiological properties, and then molecular signatures largely coming from single cell sequencing experiments. And what this has allowed us to do now, or the field to do, is to map out classes of neurons as broad as excitatory versus inhibitory, subclasses like layer-specific or specific subclasses of interneurons like the somatostatin or parvalbumin interneurons, and then all the way down to cell types. And now you could say, well, there's all this transcriptomic data available. Shouldn't we actually know whether alternative splicing operates at a cell type level, or whether it just segregates between excitatory and inhibitory neurons? And the answer is that this actually wasn't really known. And the problem is that these single cell sequencing methods are, of course, incredibly powerful for surveying transcriptomes from many, many cells and then grouping them into categories. But some of the methods like the DropSeq or 10x genomics, they don't even read the whole transcripts, and others have usually rather stochastic coverage of the transcripts. And this makes it very difficult to actually reliably assess splice isoforms. And then secondly, it's also currently unclear what fraction of transcript isoforms that you pick up by transcriptomics is actually ever turned into proteins. And so to overcome these difficulties, Elisabetta and Lisa in the lab, they adopted this conditional ribotrap approach, where you essentially introduce an HA epitope into the ribosome in a Cre-dependent way. And then that allows you to affinity isolate ribosomes, and with them, the mRNAs expressed in the cell, and especially the mRNAs that are associated and being translated by the ribosomes. And this method is as specific as your Cre line. So here they cross this with a layer 4 marker line, and then these subclass lines for somatostatin, parvalbumin, VIP interneurons, and in the hippocampus with lines for the CA1 region, CA3 region, and then also looking at the somatostatin interneurons. And then we very, very deeply sequenced this material and used a well-established pipeline for calling alternative splicing. And I should just point out that there are many pipelines out there. The reason we use this one is that it really had led to very good experimental validation of the predicted splicing changes. And now we could really analyze how similar and different cell types are with respect to their alternative exons. And what we found was really remarkable, and this is shown here. So this is a hierarchical clustering diagram. not of gene expression, but of alternative exons that are differentially regulated. And you can see that these alternative exons, so the splicing programs, really nicely segregate these cell classes into discrete categories, demonstrating very clearly that alternative splicing doesn't just operate between glutamatergic or GABAergic cells, but really down to subclasses and likely down to cell types in this sample. So as if really each different population of cells has a highly selective splicing program. This is just another depiction of that to give you an appreciation of the numbers. So between CA3 and CA1 pyramidal cells, there are over 250 highly differentially regulated exons. So really raising the possibility that this is a major driver of how you can shape the properties of these cells. Now this was exciting, but what was actually more exciting is that when you look at what the genes encode that are regulated by this cell type specific splicing, and when we looked at that with gene ontology methods, we found that it's very, very strongly dedicated to synaptic proteins. So you have essentially components of the release machinery, adhesion molecules, scaffolding molecules, and so on, that are very strongly differentially or preferentially regulated by the cell type specific alternative splicing program. And the other category that's not on here were ion channels and some components of the cytoskeleton. So really elements that make neurons or different neuron cell types different from each other. I should point out that a similar notion had been made previously for gene expression, that looking at differential gene expression, so not at the level of isoforms, and it had been proposed that this is geared or preferentially affecting synapse specification molecules, but the bias that we basically see in alternative splicing is much, much higher. So we think that a big part of why these splicing programs evolved is to indeed endow the synapses with their unique properties. So this is a very large data set, and we've actually made this publicly available in a searchable database. So if any of you are working on particular genes, you can simply put the gene name in there, and then you get plots of the differential splicing pattern across these different cell types. So to then summarize this first part of the talk, so our hypothesis had really been that splicing could be a major mechanism in shaping synaptic properties, and I've shown you one example of very highly dedicated splicing regulator, S1-2, and how this controls cell type specific alternative splicing, and then ultimately also the function of the circuit as assessed by the behavioral experiment. And I should say that one exciting direction here is actually that given that you get such a selective impact on synaptic properties, this really also highlights the potential for using splicing therapeutics to, in a targeted way, change the properties of specific neurons and the output synapses. And that's something we are very interested in. Now therapeutics also gets us to the second part of this talk. So the looking at autism-related circuit alterations, and this work by Hannah Hernberg, a postdoc in the lab. And so just as a brief introduction, I think you're probably all familiar with autism spectrum disorders. These are phenotypically defined disorders, and there are two main core symptoms. One is impaired social communication interaction, and the second is restricted repetitive behaviors. And now what makes autism, I guess, both fascinating and daunting is that there's an enormous diversity of this. So the severity of these phenotypes can be very, very varied between individuals that get an autism diagnosis. But then in addition, you also have a large number of comorbidities that are found in the patients, and some biomarkers, but it's been very, very difficult to actually attempt to stratify the autism population, particularly if you think about interventions. And I just want to illustrate this from a neurobiology perspective a little bit more. So if you simply take the phenotypic readout of social interaction and reciprocity, then from a neurobiology perspective, of course, this could arise from a whole slew of many different alterations. So this could arise from altered perception and let's say sensory hyper or hyposensitivity, or it could arise from altered interpretation and salience of certain social information. So individuals that have a different response to novelty or differ in their habituation, or for example, have increased aggression or increased aversion to certain stimuli. And clearly the circuitry underlying these different etiologies of the social interaction are entirely different, are entirely different systems. And so this is essentially the RDoC principles approach from Tom Insel. One way forward for dealing with the heterogeneity of autism might actually be to use sort of a circuit domain specific stratification. And so in our studies, we chose one domain, and that is habituation and novelty responses. And we chose this for a number of reasons. One is that there were really quite a number of studies with children with autism that showed differences in how they respond to novel stimuli and differences in how they showed habituation. This is one example here where toddlers with severe autism are shown faces repeatedly and then shown a novel face, and then you measure the gaze time that they have. And in this study, for example, they concluded that they have slowed habituation to faces, but interestingly, they actually habituated normally to objects. This is another experiment where the children showed no preference for novelty. So this is really a domain that is reproducibly affected in many autism populations. And the reason that we think this is a good entry point is that, of course, novelty exploration, as opposed to insisting always on the same thing, is really a major driver of learning. And so you could imagine that if you can intervene with that or improve that, that you can actually accelerate cognitive development. Now, to model this in mice, we use a genetic model, and this is one model that we've actually been studying for quite a long time. It's a mutant in the Neuroligin-3 gene. Neuroligin-3 encodes one of these adhesion molecules, actually one that can also in some circumstances interact with the presynaptic neurexins. And here, the added reasons why we think this is a good model is that there's actually quite a good number of studies genetically confirming an association between Neuroligin-3 mutations and autism. And secondly, and this is sort of the mouse genetics view, it's an X chromosomal gene. So, and most of these patients here are male. So if you look at male knockout mice, you're really modeling the genetic situation that you have in the human population. And there are multiple mouse models that already exist from multiple labs. And the one that we've been using is a knockout mouse where there's a transcriptional stop cassette inserted. This was generated by Rennie Hen's lab, and we collaborated with them on their characterization. But across these models, these animals exhibit a number of behavioral phenotypes that are fitting into, that are thought to be in some way relevant for autism. So they have social deficits, like reduced ultrasonic vocalizations, defects in social memory. They also have some cognitive deficits, actually enhanced spatial memory, but normal object recognition. They have some motor behavior and possibly stereotype behavior phenotypes. And then there were also some reports saying that they have sensory alterations in olfaction and visual acuity. Now, when we tested these mice in a novelty response assay, we actually reproduced data that's been previously in the literature. And that is that these mice, quite interestingly, when we confront them with a conspecific, we measure the interaction time, then we separate them, bring back together the same two mice again, and then we introduce a novel mouse. And what you can see is that the interaction time in the beginning is actually very similar between the wild types and the knockout are identical. The knockouts habituate, but then they have this very selective alteration in that they simply don't discriminate or don't recognize or don't remember. People interpret this in different ways, and don't respond to the novel stimulus mouse. And the reason that we thought this was very, very interesting is that when you do the same assay with objects, then they're actually behaving completely normally. If anything, they actually have an increased interest in the exploration of the first object. Now, I told you that these mice have a whole host of different alterations and defects. And so to really be able to conclude that this has something to do with the circuitry that is important for novelty detection, we then wanted to look at this in a sort of a conditional knockout type approach. And we focused specifically on the dopaminergic system, in particular on the ventral tegmental area, which has neurons projecting to a number of different brain regions. And by work from really a whole range of other labs, it's been previously demonstrated that these neurons get activated when confronted with novel stimuli, and that silencing these neurons blocks social but not object interactions. And importantly, also in humans, dopaminergic cells in the substantia nigra and ventral tegmental area together respond to novelty based on imaging experiments. And so the question then was whether we could replicate parts of these Neuroligin-3 knockout phenotypes by ablating Neuroligin-3 selectively in the dopaminergic cells of the VTA. And the way Hannah did this was by injecting viral RNAi constructs or microRNAs selectively into the VTA and doing that into DAT-3 mice. So we get the spatial selectivity by the injection and then the cell type specificity through the DAT-3. And when she does that and suppresses Neuroligin-3 expression in these cells, what you can see is that you replicate the global knockout phenotype. So these are these greenish lines here. The animals habituate and fail to respond to novelty. And now importantly, our knockout model also enables us to actually rescue Neuroligin-3 expression. So this is a reversible knockout with a transcriptional stop cassette. And so when we cross, these are global knockouts. If we cross this line with DAT-3 mice, we have a global knockout, but we have restored wild-type level of Neuroligin-3 expression only in the dopaminergic cells. Now, keep in mind, these are all dopaminergic cells in the whole body of the mouse, not just the ones in the VTA. But that is sufficient to rescue the phenotype. And so this really argued to us that this loss of function of Neuroligin-3 is directly playing a role in novelty detection and social recognition. Now, the next question we wanted to ask is really, how does this really work? What we focused on, or what we hypothesized, is that this could have something to do with oxytocin signaling. And the reason for that was that these mice responded normally to novel objects, but they did not respond normally to novel social stimuli. And work from many other people previously had demonstrated that in social contact, oxytocin is released and then can alter the firing in ventral tegmental area dopaminergic cells, and that this would act as a social reinforcement. And so when we treat mice, these are wild-type mice in this assay, actually with an oxytocin receptor antagonist, you can see that they pretty much look like the Neuroligin-3 knockouts. So they lose this social recognition. And so this argues that this assay really is measuring an oxytocin-dependent pathway. So what we then really wanted to ask is whether loss of Neuroligin-3 would possibly result in a loss in oxytocin signaling to the cell. And oxytocin comes from this structure here, the paraventricular nucleus, and their projections into the ventral tegmental area. Work by a number of labs previously had shown that oxytocin released onto these cells then increases the firing frequency of the dopaminergic cells in the VTA. And what made us hypothesize that loss of Neuroligin-3 could actually alter that is that, at least in studies in the cerebellum, we had previously reported that Neuroligin-3 couples to a scaffold that links to G protein coupled receptors that couple to GQ, and that their GPCR signaling was defective. So this was sort of a reason to speculate that maybe it could be important for another GPCR, for the oxytocin receptor in the dopaminergic cells. And so what Hannah did is essentially backlabel dopaminergic cells in the VTA from the nucleus accumbens. This is the population of cells that are thought to respond to oxytocin, and then cut acute slices, and then Enrique, a postdoc in the lab, recorded the cell attached recordings from these slices and perfused in oxytocin. And here you see what happens in wild-type cells. So if you perfuse in oxytocin, you get this increase in spiking in the wild-type cells. And really quite remarkably, in the neuroligin-3 knockouts, this was completely lost. And you see this here quantified over a number of animals and slices. So really the neuroligin-3 mutants, they have a lower baseline. They do not respond to oxytocin at all, whereas the wild-type cells respond strongly to this. Now, how does this work? So we know it's unlikely to be some major structural change, because we looked at the density of oxytocinergic neurons in the PVN and couldn't see a difference. We looked at the density of oxytocinergic fibers in the ventral tegmental area. And we also looked at the oxytocin receptor mRNA with fluorescent in situ hybridization in the VTA. And we also couldn't really see a difference in that. And so that was puzzling, but then we essentially started, or Hanna started, to simply do an unbiased screen to look for alterations in the ventral tegmental area. So what she did is to dissect out the VTA from knockout and wild-type mice, and then run shotgun proteomics over this. And what she observed was there were a number of proteins deregulated that are related to GPCR signaling, ubiquitin proteasome system, also vesicle-mediated transport. But what really got our attention was this cluster of proteins that relate to the ribosomes. So some of these are ribosomal core proteins, and others are translation initiation factors. And the reason that this caught Hanna's attention was that there was, of course, a substantial body of work previously implicating protein synthesis homeostasis in the pathophysiology of other forms of autism spectrum disorders. And I've cartooned this here. In red, these are autism risk genes, and it was previously largely work on RNA-binding proteins like FMRP, or components of the translation initiation machinery like CyFIP or EI4E that had been implicated in altered translation. And now we have a case where we have a cell surface molecule, neuroligin-3, where there might be a change in translation. So this was unexpected, but it was very exciting. And so what Hanna then did is to look systematically at ribosomal proteins in the ventral tegmental area. And what she found was that there was really this consistent upshift in the abundance of ribosomal proteins in the neuroligin-3 knockout. It's not a huge shift, but it's really quite a consistent shift across all proteins. And importantly, when she looked at mitochondrial ribosomes that are separate celled proteins, they do not show this change. So this really is selective to the cytosolic ribosomes. And secondly, she found that actually translation is elevated in the ventral tegmental area in the dopaminergic cells of the neuroligin-3 knockout. And this is measured here with the FunCat method, where you can see this increase in AHA incorporation in acute slices from the neuroligin-3 knockouts. Now, this was interesting, but now the question was really, how can you actually convince yourself that this is functionally important? And so we thought about ways on interfering or modifying the translation machinery to restore normal rate of translation, and then be able to test whether that change in translation is relevant for the phenotypes that I showed you before. And we focused in on a kinase called MAP-kinase Interacting Kinase, MINK. MINKs are the only kinases known that phosphorylate this translation initiation factor. And there had actually been previous work from the Sonnenberg lab demonstrating that genetic but also pharmacological inhibition of MINK restores certain phenotypes in fragile X mutant mice. And the existing pharmacological inhibitors for MINKs are not very specific. And so they actually then, this is really a multi-year effort where we teamed up with the medicinal chemist Kasum Nakro at the Experimental Therapeutics Center in Singapore. And jointly with him, we identified a new derivative of MYC inhibitors called ETC168 as an inhibitor that is brain penetrant and really highly selective for inhibiting MYCs. And so what we did then is now treat animals with this inhibitor and then ask if this would rescue phenotypes. And you see here one of those treatment paradigms. So this is a really short-term treatment, 24 hours, and then another 2 hours either before tissue isolation or behavioral testing. And what you can see here, these are data from vehicle-treated animals. These are the drug-treated animals, that this is sufficient to shift back the distribution of ribosomal proteins to its normal distribution. You can also see that ETC treatment brings back this elevated translation in the knockout, brings that back to a wild type level without having too much of an effect on the wild type, though there is some alteration here. And then maybe most importantly, the MYC inhibition also rescued the responses to oxytocin as well as the behavioral readout. So here, the open circles are the vehicle-treated knockout animals. The gray line here, this is just for reference, the wild type from the non-treated animals that I'd shown you before. And if anything, this inhibitor treatment actually overshoots the wild type response, and now cells actually respond to oxytocin again and increase their fire. And then last but not least, this down here is the behavioral readout, so the social recognition test, where you see the open circles. These are vehicle-treated animals, and the inhibitor-treated animals really recover a very strong novelty response, and there is no significant change in the wild-type animals. And this is just a small snippet of all the experiments that Hannah did on this. We also found that long-term treatments are effective. In previous work, we had seen that this rescues vocalization in the animals, and that so far we have not been able to find any adverse effects on cognitive or motor function in these animals. So then to summarize this second part of the talk, so we've added to the existing literature that social recognition in mice requires a dopamine and oxytocin-dependent mechanism. So with social cue, oxytocin is released onto these VTA neurons, and then they increase their firing through the oxytocin receptor function. Then in the Neuronal knockouts, we found what we think is a really unexpected convergence between Neuronal, this autism risk allele, protein synthesis rate, and oxytocin that had previously not been appreciated, and then demonstrated that inhibiting these MAP-kinase Interacting Kinases resets the translation machinery and then also restores function in the system. In further experiments that I don't have time to show you, we've also applied this treatment to FMR1 knockout mice, and also there find that this restores phenotypes. And I should point out there was a paper from Jill Dolan's lab just quite recently in Neuron that actually also demonstrated in FMR1 mutants defects in the oxytocinergic pathway. So this may really be a node for multiple forms of autism that one could possibly target with this. So then just to wrap up and put this back into context, so I hope I've been able to show you two examples on how these sort of transsynaptic modules shape synapse properties, and that these are not actual details of properties, but that these are really fundamental for circuit function and behavior. The first one on the Nexn1 neuroligin GluA1 link, and then in the second part on the neuroligin 3 oxytocin receptor. And with that, I just want to acknowledge the people in the lab who've done all this work. Here you have Lisa and Elisabetta. They together did this Ribotrap isolation, this large-scale effort on mapping cell type-specific splicing events. Then Lisa and Andrea collaborated on characterizing the SLAM2 knockouts and the rescue of those phenotypes. And the neuroligin 3 oxytocin receptor project was really largely, almost exclusively driven by Hannah Hernberg, and with help from a technician, Letizia, in the lab, and then with a key collaborator, Kasum Nakro at ETC Singapore. And the second key collaborator was Enrique Garcia Perez, who did all the physiology on the VTA neurons. And with that, I thank you for your attention. I'd be happy to answer any questions.
Thank you, Peter, very much for presenting us this amazing body of work. I think we have already questions, but so people can raise their hand and then I'll call you and you can unmute yourself. So there is one question in the chat, but maybe Pablo, you can, if you want, ask your question yourself.
Thank you, Gaia. Great talk, Peter. Thank you. So I was wondering, I don't know if I missed it, but have you seen a huge improvement with the inhibitor of MINC? So have you tried a combination of both treatment, oxytocin and the M&K inhibitor, to see if there is a summatory effect in the behavior?
No, we haven't done that yet. So our interpretation would be that you may amplify the responses, but we haven't tested that yet. And what we have done is the converse. We've combined the MINC inhibitor treatment with the oxytocin receptor antagonist to convince ourselves that that behavioral rescue is still oxytocin dependent and not driven by something else. And they're as sensitive to the receptor antagonist as the wild-type animals are. But my view on the, I mean, there is a lot of clinical trials on intranasal oxytocin application in patients, right? And they seem to have rather varied conclusions. And I think I think it really depends on what type of phenotype or situation, what kind of group of individuals you're looking at. But in our model, we would think it's really the reception of the signal. So providing an abundance of exogenous oxytocin may not even be beneficial in that type of situation, right? But it would really be about the amplification of the endogenous oxytocin signal, which would have the benefit that it remains tied to social context.
So next question, Andrew, can you unmute it?
Yeah, I can unmute. Hi, Peter, it's a lovely talk. I have a question about this really nice reversible loss of the LTP, where you get rid of the splicing factor and you lose LTP totally, and then it comes back. I'm just wondering I'm not sure if you mentioned it, but what do you think is happening on the level of individual synapses or synapse number or synapse content? I find that result absolutely fascinating.
Yeah, so we know that synapse number is not changing. We know that if we stimulate the shared collateral fibers, that evoke transmission is elevated, but a little paradoxically, the mini amplitudes, as far as we've observed them, are not altered. So we don't completely understand how that exact phenotype comes about. What we do know is that the biochemical interactions between the Nexn1 splice variant that's now expressed in the SLM2 mutant, that variant doesn't interact with certain post-synaptic proteins anymore, like the neoligin 1, for example. And so that makes us think it's a transsynaptic coupling that's altered. And we hypothesize that when you give an LTP stimulus, that new AMPA receptors may actually be put on the surface. They're actually too many to begin with, but that they cannot be tethered, they cannot be incorporated in the synapse. But I think this would require really much more sophisticated measures, possibly surface tracking of receptors to really quantify and assess this properly. Does that answer your question?
Oh yeah, very much. But maybe I can just follow up. I mean, there's an alternate hypothesis that the synapses themselves are actually different, that they're un-LTPable because of a different structure altogether, that they have different sizes, or the kind of things that you could see by EM or tomography or I mean, there's a lot of possibilities. I just find it so interesting.
Yeah. So we have not done that much structural work on this. We've looked at sparse GFP film, and there with conventional confocal microscopy, we haven't seen size differences. But obviously, this doesn't have the degree of resolution that would be needed to really look at the types of parameters that you're bringing up. Yeah.
Thanks very much. Thanks.
Pierre?
Yeah.
Go ahead.
Question over here. Thanks a lot for the great talk, Peter. So, yeah, I have many questions actually, but maybe just focusing on one. So I was struck actually by the graph that you showed, on your first slide. I wasn't aware of this, the difference in number of splice variants between primates and mice, so even between mammals. And so in that sense, I was wondering, have you or others tried to look at splicing diversity, in a subtype specific way, like you've done in the mouse, but using human neurons, reasoning in particular that, the jury is still out whether in the adult there are more or similar number of neuron subtypes in the human brain versus other brains. But I think I would argue that for sure during development, you may need a more complex set of synaptic, synaptogenic or synaptic specificity molecules and their transcript variants. So can you comment basically on this difference of splicing diversity between species, and do you have any hint of what could be upstream? I mean, is there a different level of or diversity of splicing factors? That's my question.
Peter, you are muted.
Thanks. Sorry. So a lot of this has come from work from looking at bulk sequencing data. And there's another, I think, equally fascinating thing, that if you look at organ-specific exons, then they're actually, they have sort of separately evolved, meaning that the organ-specific exons they're more similar to the species that you're in, whereas the gene expression in the organ is if you have the same organ from different species, they're more similar to each other. So there has been some very rapid evolution of organ-specific splicing that has occurred separately. And this was discovered about 8 years ago between Chris Burgess and Ben Blanco's lab. And it's something we're super interested in. And it would really now, the problem is that unless you're the Allen Brain Institute getting enough and sufficiently deep SmartSeq sequencing data to really be able to reliably call splicing, you need to use an alternative approach. And that's why we sort of sidestep that to then use this Ribotrap approach. I think we actually once talked at a conference whether one could put that into human neurons and then with the emerging enhancers drive this in selective subpopulations. And I think that there's a huge amount to explore and it's super, super interesting. The other finding that we've had was that between cell types, the transcription start sites were highly different. So half of the diversity distinguishing neuronal cell types are alternative transcription start sites. And that hasn't even been that detailed analyzed to my knowledge across in the human or in the evolutionary perspective. But that's a whole other level of species-specific gene regulation that's exploded, and that I think would be a main driver for phenotypic diversity. Absolutely. But to my knowledge, none of this is done because people do drop seek, right? And you don't see any of this, right?
So, yeah, I mean, indeed, maybe I guess you would have to think thoroughly about, what would be the right driver. But indeed, I think an exciting experiment would be to try to do ribotag in a population that is reasonably well defined. Now, of course, I realize this is not an easy definition, but that would be ortholog, between mouse and human, for instance, and then just see what's the splicing diversity looking like, because you would assume that this might be true for many different types of neurons. You don't necessarily need to go into complicated cortical interneurons and so on. My other related question was, do you have a sense of what could be upstream of this? And maybe in line with this, you started by saying there are many more splice variants where there are similar number of genes, but the main example that you take, I mean, of course you're aware of this, shows remarkable specificity between the splicing factor and the splicing events. So then you're back to the same question, how many splicing factors do you need to get this diversity? And you would think that probably you would need factors that are acting to generate more diversity. So do you have a sense of, could there be families of splicing factors that we don't know of yet? And
yeah, so we're the two. I think that from the RNA binding protein perspective, I think one important thing is that we have good evidence that some of these splicing factors act orthogonally to the cell lineage, meaning it's not like that you basically, that all glutamatergic neurons use one pool of splicing factors and then drop or overexpress one or the other, and the GABAergic neurons have a different class. We've examples where, for example, SLM2 also regulates certain exons in a subpopulation of somatostatin interneurons. So that gives you a lot more degrees of freedom to regulate this. Second, there is very good evidence for synergistic action of multiple factors, density of binding sites near the representative exons that are being regulated, and thereby recruiting and changing the dynamics of the splicing. And there was something else I wanted to say. Yeah, then there is a whole body of literature now. I don't know if any of these will emerge as splicing regulators, but the Hensel lab, Matthias Hensel's lab, right, has done this unbiased proteomic screens for RNA-binding proteins and discovered a whole range of novel RNA-binding proteins that do not show any resemblance to common RNA-binding motifs. So theoretically, there would be an enormous diversity out there. While I'm answering this, I'm actually just saying that Manuele Rimea is on the seminar as well, who probably knows a lot more than me even about all of this evolutionary stuff. So I don't know if you want to say something to this, Manuel.
Manuel, if you want, you can.
Thanks.
In the meantime, maybe I can ask questions also related to that. So you're saying that, so you were writing that the end alternative splicing is important for terminal differentiation. So imply, if I understand well, that it's kind of the fine tuning of the neuronal network. But I was wondering whether you could argue that those alternative splicing events maybe have even stronger impact in really kind of determining, changing even cell fate at one point. Do you know if you can push one cell to be another cell type just by expressing different splicing forms?
Yeah, so we have focused on factors that are sparsely expressed and that are selectively expressed in cell populations. And there, we think they're really more like terminal selectors. There was actually a paper from Xiaodong Fu's lab that was published in Nature, I think a few months ago, where they showed that, I think it was a PDBP knockout, where they could basically shift astrocytes into dopaminergic neurons. So very early sort of cell fate shift and switch. I would expect that many of factors that have that type of potential or capacity would be factors that are regulating much broader sets of target mRNAs and then really a more coherent program. But that's sort of speculation. And then Chris Walsh's lab had actually also profiled early alternative splicing events in neuronal differentiation, pre and cortical development. And there's certainly actions like that. But the ones we've been, in a way, really try to focus on this sort of terminal differentiation program. And so the kind of statement that I made applies to those, but there's certainly other modes how the splicing can act.
Yeah. Good. So I don't see any other question from the audience. I think we are slightly over time. It doesn't matter. But so if there is any other question, please raise your hand now. Otherwise, I'm gonna close here. And I would like to thank you again for the excellent talk. Hopefully see you around at one point again in person.
In real life, yeah.
In real life.
Thanks everyone for
Thank everyone for being here, and yeah, great talk. Thank you.